Hi everyone, I'm Sagar from Sri Venkateshwar College of Engineering. Today I'm going to discuss about DNA fingerprinting in forensic investigation. Let us start with the introduction first. DNA fingerprinting is a powerful tool used in the solving the crime scenes. By analyzing the unique patterns in the individual DNA, we can determine whether the person is present in the crime scene or not. By taking the individual patterns of the DNA, we can identify whether the person is present in the crime scene or not. It can be determined using the forensic DNA fingerprinting. In this presentation, we are going to discuss more about the science behind the DNA fingerprinting. Now let us know about the history of the DNA fingerprinting. The DNA fingerprinting was first developed by the Sir Alec Jeffreys in the year 1984. He discovered that certain regions of the DNA is variable from one individual to another individual. By using that certain individual, we can identify the genetic profile. We can create a unique genetic profile. This led to the development of the DNA fingerprinting for the forensic tool. Now let us know about the basics of the DNA. You guys all know about the DNA stands for deoxyribonucleic acid. It is a complex molecule that can contain all the genetic information required for organisms to develop and function. It is a basic complex molecule that is required for an organism to develop and functioning of an organism. DNA is made up of 10 nucleotides, that is the 4 nucleotides. It is adenine, thymine, guanine and cytosine. These nucleotides pair up together to form the ladder-like structure called as DNA that you are seeing in the figure, that is DNA structure. Now let us know how DNA fingerprinting works. DNA fingerprinting works by the analyzing the short tender repeats. It is called as a STRs in the DNA. These are region of the DNA that can vary length between the individuals. The length of the STRs is varies from one individual to the another individual. The DNA is extracted from sample and amplified using the technique called a PCR. From the PCR technique, we can amplify that DNA samples. The amplified DNA is then separated using a technique called as a gel electrophoresis. From the gel electrophoresis, the patterns has been separated. From this gel electrophoresis, this gives the separate unique bands of the pattern. From that we can differentiate one individual to the another individual from this gel electrophoresis technique. Collecting the DNA evidences. Collecting the DNA evidence requires a special prevent of contamination. The sample must be prevented from the contaminations. The samples can be collected from the blood, semen, saliva and other body fluids. DNA can also be found in the objects that have been touched, such as cloths and weapons. Once collected, the DNA is extracted and can be analyzed. Let us see the DNA profiling. DNA profiling is a process of analyzing the sample of DNA to create a DNA of a unique fingerprint. It is used to create the unique DNA fingerprinting. This fingerprinting is created to identify the regions of the DNA molecule that vary between the individuals. This resulting profile can be used to link the suspect from the crime scene or exclude the innocent individual from the suspension. It is used to link the suspect to the crime scene, or we can exclude the innocent person from the crime scene by using this DNA profiling. Now let us see the limitations of DNA fingerprinting. Although DNA fingerprinting is a powerful tool in the forensic investigation, it has some limitations and it requires a high quality of DNA sample. From the low quality we cannot identify the DNA sampling or DNA fingerprinting. It was too difficult, which may not always be available. It can also be subjected to the human error, contamination. Additionally, the DNA evidences has been challenged in front of the court and interpretation of the DNA results in the complex. Now let us see the applications of the DNA fingerprinting. DNA fingerprinting is a wide range of applications in the forensics, including the identifying the suspects in the criminal investigation, exonerating the innocent individual, that means accepting the innocent individuals, identifying the victims in the mass disasters and establishing the paternity. It has also been used to solve the historical mysteries such as identifying the Romanovs. Now let us see the summary of this presentation. DNA fingerprinting has revolutionizing in the way of investigation and solving the crimes. By analyzing the unique patterns in the individual of DNA, we can link the suspects or to the crime scenes or exclude the innocent person from the suspension. While there is limitation is to use, DNA fingerprinting remains a powerful tool in the law enforcement and the justice system. And that's it for today's presentation. See you guys in the next presentation. Until then, take care.